Hello and welcome to Coast and Country, powered by the science of the Connecticut Agricultural Experiment Station. Coming up in this special edition, all about Plant Science Day 2024. We talk with Brian Herbert, Commissioner of the Department of Agriculture, and the Lieutenant Governor, Susan Bysewicz, about the latest developments in the ag industry here in the state. Felicia Millett shows us how to graft tomato and potato plants together to get what's called ketchup and fries plant. Jeremiah Foley shows us how to prevent the spread of aquatic plants in our waterways that are considered invasive species. And we catch up with members of the public as they get their plant questions answered. But first, we catch up with Dr. Jason White, Director of the Connecticut Agricultural Experiment Station, and his opening remarks on Plant Science Day 2024.
So as you can see, we were established in 1875, and if you do the math in your head, that means next year is our 150-year anniversary. So we'll be having a number of events over the course of the year next year to kind of celebrate that landmark. As noted, we're the first and the oldest agricultural experiment station in the country, so I think 150 years is something clearly worth celebrating. Because it's a question that always comes up about money and operating budgets, I just put that up there so that you can see. About 56% of our funding for our operations comes from the state of Connecticut, but the rest is federal. And comes from a series of sources, pass-through funding, but also competitive grants. A fair number of our 50 or 50 plus Ph.D. scientists, their job is to work on research topics to get funding to support that research. So a lot of that funding that comes in is competitive funding. As you can see, our primary mission, it's in 4 core areas. I think most people are familiar with these: agriculture, environment, food safety, and public health. All of those core areas, plus our outreach programs, are represented today. So if you can brave the weather and the rain, you can see all those. Research is our primary output. That's really our main focus, but we do have a number of statutory responsibilities. So these are things that we're required to do by state statute, and as you can see, those are listed there. Everything from inspecting plants and for pests in nurseries, to analyzing food, to cannabis, the tick testing laboratory, the mosquito surveillance program. All of that is statutory work that we do. And then I mentioned our 5 departments, which you see there. In terms of updates to programs, I just picked 2 to focus on because they're kind of the 2 that have been in the news most recently, part of our vector-borne disease program. The first is just an update on some of the things that are happening in our tick testing program. This, like the mosquito program, it's a combination of statutory work funded by the state, pass-through funding from CDC that goes to the Department of Public Health, and then direct funding for research that comes from the CDC. So I think most of you have probably heard me talk about this multiple times. The biggest issue we're having with ticks is the new invasive species that are coming in as a result of the changing climate. And and those new species carry new pathogens, pathogens that we're not familiar with, and that's one of the biggest public health issues we're facing right now in the state. So you can see that we just put the numbers up there just so you know. In the last year, we've had over 5,700 ticks that were submitted for testing. Of those, we test about 4,100. We have criteria to determine which ones go forward for testing, but basically it's if they had a blood meal. And we screen for a number of organisms. It was expanded this year to include these 2 other organisms, Borrelia and Powassan virus. These are 2 of the emerging ones that I was talking about. But some of the ticks that we're seeing more and more of are these invasives that are pictured on the right there. There was a case of a human disease that was actually linked directly to one of these invasive ticks. That's this disease that's related to Rocky Mountain spotted fever. So this could be a big deal because it's a direct interaction between these invasives and human disease. So that program is extremely active and will continue to be so. We used to say we had a tick season. We don't have a tick season anymore. We get tick submissions in January and February as well. So It's one of our most important services. The other is the mosquito program. Obviously, if you're listening to the news, you're hearing significant West Nile and Eastern equine encephalitis activity this year. That's going to continue throughout the summer. But again, this kind of just highlights some of the program updates. We have 108 traps that we set up throughout from May to November. And the idea behind this program is that if we find disease in the vectors, in the mosquitoes, that's a leading indicator for what's going to show up in the human population. We have historical data showing that that's true. So this program is really important because it shows us what's, it gives us a 2 to 3 week heads up of what could be showing up in human populations. And again, you can kind of see some of the updates that we have. And we've got some exciting new research initiatives as part of this CDC Center of Excellence that we're involved with Cornell University and some other schools. And then 2 other things to kind of focus are our big construction projects. For those of you who have a good memory, this slide looks shockingly similar to the one last year that I presented. But we have made some progress. Our Valley Lab renovation project. So our main location is in New Haven, but we have a location up in Windsor, which we call our Valley Laboratory. The site was established in the 1920s, but the existing structure was built in 1940 and hasn't been significantly renovated since. Wrong button, sorry. I don't know if you can see my highlighter, but the existing structure is there, and on this schematic, that's the structure there on the left. So it's a significant renovation of the existing structure and then construction of attached state-of-the-art laboratory space. So it's a $36 million project. We had a kickoff meeting last week. We're moving into the construction document phase. The ball is rolling, it's going downhill, and it's not going to stop. So we're looking at probably a groundbreaking at some point in late summer, actual shovels in the ground late summer or the fall. So that will be very exciting for us. And then the other project that's happening right now in New Haven, just started this week, is renovation of our greenhouse facilities, which is about a $1.6 million project. So that's all I have. I just want to thank everyone for everybody who was on the Plant Science Day Planning Committee. Plant Science Day, it's kind of like painting the Golden Gate Bridge. The planning for the next one basically begins the day after the previous one stopped. So it's a significant amount of effort in terms of putting this together. But I think the thing to keep in mind is really it's just a way for us to showcase what we do for the citizens of the state, how we bring science to the benefit of the community.
So for any of you who are not familiar with the term grafting, grafting is basically taking a part of one plant and combining it with another part of another plant. You can actually do this with several different types of plants, combining them and making one single plant. So one rootstock is supporting however many grafts you've joined on top. So the basic terminology, the part of the plant that is below ground is called our rootstock, so that's really easy to remember. The above ground portion is called the scion, S-C-I-O-N. So the part that has the stem, the leaves, the flowers, the fruit, the part of interest is the scion, or the top plant. So in this case, this is more of a novelty project. This is not something that's done widespread in agriculture to try to, make the most of your planting. This is just something to do for fun, but also it's a great educational experience to try to do this at home. So with grafting, there's a few important concepts to keep in mind, and if you ignore these concepts, you probably will have a lot less success with your grafting. So the poster that Joe's presented for us explains basically how the grafting works. So in our plant's vascular system, or in the stems of our plants, we have different elements, which actually on the illustration, this is an illustration of a woody plant cross-section. Actually, our tomatoes are herbaceous dicots, so it's a little similar. There's a few differences here. But the really important thing I want to explain is that for that upper plant to receive water and nutrients from that rootstock, we need that vascular system to connect, to fuse together. And there's one part, we always see the growing points as plants extend in height and in width, but actually the stems have a secondary layer of cells that grow and increase the diameter of the stems as they go outward. And in our plant stems, this is called the cambium. The cambium layer in the stems is where new cell growth occurs and where cell division is happening. And it's very important, even if you forget the name of it, for the cambium layer of your rootstock and your scion to join together. This is going to establish the fusion and strengthen that graft. It's going to callus, it's going to eventually allow the vascular systems to join, and without that fusion, your graft will fail and your scion will just wilt and fall off. So it's very important to join these two areas. So an easy way to remember that is you're going to have more success with your grafting on a very basic level if your stem diameters are the same. So actually with tomato grafting, you would be doing this much, much earlier in the season. So the plants I have to show you, their diameters don't quite match. So it's probably going to be a little bit lower success rate than what we would hope for. But ideally, if you were doing this on your own, you do this in the springtime with plants with matching diameters. Also, earlier in the spring, you're more likely to be able to grow that plant to the correct harvest time. This is a bit late in August, but it's Plant Science Day, so we're just going to show you the technique here. So another thing that will determine the success rate of your grafting is going to be the relationship between the two plants that you choose to graft together. So actually, with grafting, a lot of times grafting is used in ornamental horticulture or in agricultural production, usually for things like improving some traits, either of the appearance of a plant or higher yields, disease resistance, things like this. And most of the times they're done within the same species. So for tomatoes, we might do grafting to avoid certain diseases. And this is going to work if the diseases are in the soil. Things like verticillium, things like fusarium. There's a lot of success with grafting tomatoes to a rootstock that is more resistant to those soil-borne diseases, and then taking the scion, or the top plant, of a species, or a cultivar that has a desirable trait, perhaps something that is a fruit type or the tomato type that you prefer, either for its marketability, its color, its flavor, and joining it to a rootstock that is less likely to get a soil-borne disease than that desirable cultivar that you wish to harvest. So that's a major consideration with tomato production. It can also be done, I think, in some parts of the world, like Mediterranean and Israel. There's a lot of salinity in the soil. That's a very hard thing to get rid of. So they might find rootstocks that are better with growing in that high salt content soil with the cultivars or the varieties that they want grafted on top. This will be done in a situation where the desirable variety just doesn't have the ability in its rootstock to overcome some of those challenges. So those grafts tend to be very successful because they're within the same species. So if we're grafting a tomato to a potato, they're not the same species, but they are the same genus. So if any of you are familiar with botanical terms, a lot of the times we use two botanical names for most of our plants. And actually, both the tomato and the potato are solanum. So does anyone know what the third very common vegetable plant with solanum? Yeah? Eggplant? Oh, pepper, not pepper, but yes, eggplant. Eggplant. So I don't know. It makes me think this might be something you can do with eggplant as well. I haven't heard of anyone doing that. But this gentleman who tried to do a cucurbit with a solanaceous plant, it's possible the graft didn't work because they're too far apart in their relation to one another. So the closer they are, if they're in the same species, so if you have two types of tomatoes, they're different varieties but same species, the graft should take. In this case, we have two different species in the same genus. There's a slightly lesser chance of success, but it will still work. So the materials that you need for your grafting, this would be the same if you're doing just tomatoes to tomatoes, but in our case, tomatoes and potatoes. So we need our tomato scions. So these were all grown from seed in the greenhouse in New Haven. And then our potato plants, which were grown from seed potatoes, which are just those potatoes that are sprouting. You'd cut them into small sections, let them callus, and then those are kind of lower in the pot. It's very important if you are doing anything in containers, to make sure you have drainage holes. So I'm not going to lift this, but trust me, there's a lot of drainage holes because potatoes can actually rot if they are sitting in water that's not flowing through. So we've got our plants here. We've got grafting knives. I've got a couple of hand pruners. For tomatoes, they're so thin and herbaceous, I'm just going to be using some straight razors for these. It's great because they're inexpensive. We can clean them and we can just throw them out after a couple of grafts. This is protective finger tape. It sticks to itself. I didn't cut my finger yet, I swear. This is just a nice thing to have, except this one isn't sticking to itself. Box of Band-Aids. What else? Stakes, because I'll show you, the stakes will make sense in a little bit. And some plastic bags, a spray bottle. Okay, so the first thing we're gonna do is on our potato plant, and we wanna work quickly, we don't want things to dry out. We're going to find a stem, like this one right here, is a little closer to the diameter of some of our tomato stems. And so we're gonna remove the potato. And on a plant, so remember, this ideally would be done on plants started indoors early in the spring. So they're gonna be a lot smaller than this when we do it. We're gonna take our potato plant, and on your seedling, you would go below the cotyledon level. The cotyledons are those first leaves, leaf-like leaves that come out of the ground. They don't look like the true leaves. They don't have the serrations or the lobes that our true leaves would have. It's very important to cut below that when you're starting with your young potato, because, and I'll explain a little bit more later, but we want to discourage the potato from sending any new shoots. So we're going to cut this down pretty low. It's going to be a little bit hard to see. And I'm going to cut at a 60-degree angle. Some places will say a 45. So this we don't need anymore. And then the same on my tomato, which this is already a little bit too old. It's about twice the diameter that it really should be for this stage. But again, on my tomato, I'd also want to cut below the cotyledon, which is no longer attached on this plant. And that's a 60-degree angle as well. As long as I make sure my stem of my scion and my rootstock are lined up together, I can wrap this. The nice thing about the grafting clips is they're gonna fall off on their own, so you don't have to worry about them cutting into your stem and girdling the stem or doing any damage. As that stem expands, they just pop off. They're like a very weak spring. This saran wrap you would have to remember to pull off on your own because the plant won't be able to remove it. All right. A little bit of support here. All right. I'm going to take another stake. Okay. Now what's really important is we want to cover this whole system with a plastic bag. And the stakes are really there both to support the scion of the tomato, but also we're going to use it to hold up this plastic bag. And we can spray the inside of the plastic bag with a little bit of water, but don't just spray the tomato leaves because that can, if there's any kind of fungal issues, you might exacerbate some infection with that. So just spray the bag itself. And then leave that 24 hours in the dark, and then after 24 hours you can give it indirect light. It should take anywhere from 3 to 5 days, maybe a week for the graft to take. It really depends on your plants, the temperature and humidity in the area where you've done this. I think our recommendation is about 75 degrees Fahrenheit, as close to 100% precipitation as you can get.
It's important that, as we think about our backyards, we also think about the impact on Connecticut's farms. So last year, rain all the time. This year, it was very hot and humid, sporadic thunderstorms and rainstorms, but now we're getting to a point where I'm getting pretty nervous that we're getting too much rain. The ground's saturated, the rivers are swollen, but I think it's indicative of a changing climate, that the weather events are more unpredictable, but also more intense. And so how do we help farmers manage through these changes so that they can get a crop every year regardless of what Mother Nature throws at us? And a lot of people think of the Connecticut Department of Agriculture as kind of this multifaceted U.S. Department of Agricultural organization. But we're really not. We don't do science and research. That's the Connecticut Agricultural Experiment Station, that's Yukon Extension, that's where that happens. And so what we count on as our friends and partners at the Ag Experiment Station to develop the technology, to understand what needs to be done to prepare the plants for this changing weather and make sure that we have good crops or good varieties for farmers to take advantage of. And then our role at the department is to help them implement that. And that's where we can use some of our grant programs to reduce the burden or the cost of entry on a transition to a new crop. We're putting more money out, investing into more farms, and we're also diversifying the farms that we have. We have to acknowledge that agriculture in 10, 20, 30 years is going to be a little bit different. So we have to encourage the diversification. That means increasing the diversity of who's growing, increasing the diversity of what they're growing, the diversity of where they're growing, how they're growing, and then the value that they're selling it for. And so farmers are looking at us to help them with that process. And so if you look at our program, our grants programs over the past years, we've been able to tick off on all those boxes to make sure that Connecticut's farmers today have a profitable operation to sell to the next person or pass down to the next generation. We may not be happy with the rain right now today, but we get rain. And that's really important in the ag space. And when you look across the country or internationally, there are places across this world that are not getting rain, that are in severe drought. So it becomes really important that we can continue to grow crops to support Connecticut's population and our neighbors, because they may not be able to do it themselves.
Our agricultural sector is really important to our economy. It employs over 20,000 people and is responsible for $4 billion in gross product for our state. And I said it's also important for the community because, as you see, there are hundreds of people here, even though it's raining, because they're looking forward to connecting with the various parts of the agricultural experiment station and getting their questions answered. But community organizations that have to do with farming and agriculture are here as well. So it's really a great day, and I'm hoping the sun's going to come out for everybody.
We're really proud to have the Agricultural Experiment Station in Hamden, and it draws a big crowd every year. I wish we could come out here more often. And it's a really big piece of our community. So a lot of the community organizations that come here and connect, so many people here on a dreary day. We have the Sleeping Giant Park Association celebrating their centennial this year. We're just really proud to partner with the state for this beautiful piece of property and all of the community organizations that care so deeply about the environment.
And I'll tell you that just as a resident of Connecticut, I have been helped by the Agricultural Experiment Station. I had 2 flowering trees that were diseased, and I was able to send pictures to the experiment station. Unfortunately, I had to cut the trees down because there was no way that they were going to come back, but it was nice to be able to have that resource that people can use, and thousands of Connecticut residents do, but I want the people of Connecticut to know what a precious resource this is.
My background is in engineering, so science and engineering is really important to me, making sure that the best and brightest minds are moving forward. And we have so many different programs in our schools to really elevate women in science and engineering, and I think it's fantastic that this exists in Hamden.
Well, and I'll also add to what the mayor is saying. We are seeing in Connecticut that the newest group of farmers are women. And women who don't come from a farming background but appreciate the ability to grow food in their backyard, in window boxes, and they have started some really great small businesses. And our state agricultural department is here to help, along with the experiment station. And also the mayor and I have had the opportunity to meet a lot of female and male scientists who are here doing some of that important research that I talked about.
Picking up a lot of information for the Wallingford Garden Club.
I'm Jeremiah Foley. I came up here from South Florida last year, and I was hired as a new scientist for the Office of Invasive Aquatic Species. Now, this is a continuation of the lake surveys that we were already doing since early 2000s. And so this office was formed because of a new threat to our Connecticut rivers and water bodies. And that's this plant right here. This is known as hydrilla. It gets its name from Greek mythology, the hydra, because it has so many heads. You cut it off and you get a new one. So before I get too down into the weeds, pun intended, I want to sort of take a step back and talk globally about what biological invasions mean and what they do. So biological invasions are, I'm sure many of us are familiar with, plant, an insect, animal gets brought here from another part of the world, and it establishes and doesn't particularly have any natural enemies and can start growing and wreaking havoc. Now, the United States spends about $15 billion a year managing or dealing with invasive species. And in the cost of treating terrestrial systems really outcompetes the cost of aquatic systems. That's except for New England. New England, our cost of managing aquatic systems from invasive species outpaces that of terrestrial ecosystems. So here in New England, and in Connecticut in particular, we consider Connecticut the gateway of invasive aquatic plants to the rest of New England. And the reason I say that is because in Connecticut, based on the surveys we've done, about half of our lakes and rivers and water bodies has at least one invasive species. So that's about 50%. The baseball card stats of this plant are really remarkable. So this plant can grow 191 inches a day, and everyone's like, oh my goodness. But that's not to say a single strand is growing 191 inches, but a single plant can have 191 tips, and each one of those tips grows an inch a day, and you can have hundreds of thousands, if not millions, of plants in the environment. So the rate of biomass accumulation is phenomenal. This is a free-floating plant, meaning it's on top of the water surface. This one is more cryptic. It's in the water column. It fills out water columns. Another shocking baseball card stat with this is you can manually remove these plants pretty easily. You can pick it out and you can put it on your kayak and get it to shore. This, if you go to remove it and you create 3 new fragments, each one of those fragments then becomes a new plant. And so you can imagine what a prop does to a population of this plant. If you get stuck in it, not only can you not get through it, but now you have increased the number of fragments, and those fragments then can be the genesis of new populations. Historically, hydrilla has been in the United States since the 60s. There's 3 strains of hydrilla. We have one that was introduced in the 60s, and it was found planted really nicely in these nice lines in someone's pond because they were using it for the aquarium trade. So oftentimes these plants come here from other parts of the world because of human-mediated activities, but specifically ballast, and second to ballast can be the aquarium trade. So in an aquarium, you like to find plants that are easy to grow, resistant to insects, resistant to disease, and all those attributes make it really great for an aquarium. But when someone's done with their aquarium, they tend to just dump it. And we've found whole aquariums in water bodies before. So it's not just something people say. We've actually found full aquariums inside water bodies. So the aquarium trade is really getting the connectivity and finding those plants for these enthusiasts is really getting the plant from one location, say from Asia, where hydrilla is, to here. So we have one strain that was introduced in the 60s. Another strain popped up in the 80s, and that one is found near Delaware. Then in 2016, the Connecticut River was ground zero for a new hydrilla biotype or subspecies. Now, The two that have been here since the 60s and 80s are more genetically similar to one another than this third strain. This third one shares a different clade, a different common ancestor, and it looks and behaves differently than those other two. So this is a big question mark on how to manage it compared to the other two, but we got a good book of knowledge based on those other biotypes that have been in Florida. So once a plant gets brought here, okay, it makes sense, aquarium, but how does it get spread from lake to lake to lake to lake? When I got here to Connecticut, they said, Connecticut River hydrilla is only in the Connecticut River. And that's what sort of this hydrilla monster looks like. But since last year to this year, we have now found it in 9 new locations. And we found it next to boat ramps. And we're finding it at small populations, suggesting that it's recreational boating activities and they're small populations. But if anyone knows biological invasions, you know the way this progresses. It's this slow, it's establishing, and then there's this exponential point where it takes off, where the number of sites where this plant is, or an invasive plant, starts to really go through the roof. And that's where we're at now with hydrilla. The best way to go about preventing the spread of invasive aquatic species is this type of outreach education, but also slow the spread, stop aquatic hitchhikers, because this is how the plant's getting around from location to location. There's a simple clean, drain, dry. It's pretty self-explanatory, but there's a couple nuances to it that people might not realize. So clean, what does that mean? You can use a pressure washer with biodegradable detergents or a mild bleach solution with a scrub brush, and you take your boat out of the water, and oftentimes a lot of the aquatic plant material is on the trailer, not necessarily the boat. The boat can have the prop fouled and everything, but the trailer can also serve as a significant holding source for aquatic plants. And it's pretty striking to see a boat come out of the water and it just looks like snot, for lack of a better word, is all throughout the plant, all throughout the trailer. Another aspect to this is the hidden nooks and crannies of a boat-trailer interface. So when you get your boat out of the water, oftentimes between where the boat is laying on the trailer and the boat itself, you can get these type of microenvironments where they are pretty much locked in and sealed. And you think your boat's clean, but there can be these hidden compartments. And there's studies have shown that you can take a plant, and they did this with hydrilla, you can take a plant from Florida and 24 hours later, you can drive up to Canada and it's still viable. What was really cool about one of those studies is that, and this was counterintuitive to me, is that those boats that have, say, a fabric on them, do a really good job of wicking that moisture out of those environments, as opposed to plastic on plastic or metal on plastic type of deals. So that was news to me. So it's really important to clean your boat, pressure clean it, and then you have to drain your boat. That's to empty the bilge, empty your boat, your bait tackle, and even your fishing lures. Not just your boat, but your fishing lures, your nets, all of the equipment that you use should be cleaned and then drained and then dried. Now, drying isn't just like, okay, I'll drive down the road as fast as I can and it'll be dry. Drying takes a while, especially if your cabins aren't open. So the recommended amount of time for a drying is 5 days. But that's a lot, that's hard, especially if you're, say, in a fishing tournament where there's competition coming on, right? So you gotta be from one lake to another, you're competing to win, you got 40 or 50, maybe 60 people within that competition circuit getting up and moving. So our data has shown with all these lakes that now have healthy populations of hydrilla, they're tightly correlated to fishing activities. So we were finding new locations of hydrilla solely based on circuits. So it would be like, okay, let's go to this circuit and let's go to this lake and let's look, and we would find it. So I'm not trying to target Bass Pro fishermen or any fishermen in particular, but I think it's an artifact of competition. Spin it now towards understanding hydrilla and what we are going to do as scientists, land managers, to understand how to start to combat hydrilla. We're teamed up with the Army Corps of Engineers, like I mentioned earlier, And they are here in the Connecticut River now. Actually, today, they're putting in herbicide applications in a select few coves to demonstrate what herbicide to use, when to use it, and how to use it. There's a lot of misconceptions about aquatic herbicides and how effective and specific they can be. So for all the hundreds, if not thousands of products approved for release in terrestrial ecosystems, there's a handful, 12, 13, 14 maybe, strictly regulated herbicides through the EPA that can be used in aquatic ecosystems. So knowing how to use them, when to use them, and where to use them plays a big role, especially in a system like the Connecticut River, which is hydrodynamically complex, right? There's flow, right, which is problematic in its own right to try to keep a product, but it's also tidally influenced. So it's really, last year with the Army Corps, we characterized the flow dynamics of these sites that we're now treating this year with herbicides, and we're using a handful of products that have really remarkable chemistries. Oftentimes people have this idea of when you put herbicide in the water, it's everywhere, and that's not the case. Herbicides can be quickly bound up by the plant material itself. They, depending on the product you use, 24 hours later it can be completely degraded to its basic elemental parts, and light also breaks down the plants. The products rather. So we've put the herbicides in the water at a select few sites. Some of you might know Keeney Cove, Selden Cove. There's these really pristine habitats that are completely inundated by hydrilla. You get out there and it literally looks like this. Like there was open water body, and it's completely, the water column is completely filled in with hydrilla, and it's wreaking havoc on not just the ecology, but the economics of the region. So the way this plant reproduces is through two mechanisms. It flowers, but the genotype we have here is only the male flowers. There's no female flowers here. So it's not sexually reproducing. Well, how it's reproducing are these things we call turions, which are effectively like a bud to a plant. You know, the daylights get shorter. Instead of elongating, the plant starts to pile in on itself, those inner nodes, and that becomes essentially the bud that serves as a seed bank from year to year. Those things break off at the end of the year. This plant has a wicked tendency to auto-fragment, so it gets towards the end of the season and it breaks itself apart. And all those fragments can now drift and start new populations. There's another aspect to not this hydrilla, but the others two in South Florida, the dioecious and the monoecious in Delaware, where it produces these things called tubers. And while it's botanically not a tuber, for all intents and purposes, it's like a potato-like structure that is in the hydrosoil of your lake water bottoms. So that's effectively the seed bank. This plant's not producing those things we call tubers. So there's a silver lining here in the sense that the seed bank might not be as persistent and as intense as those other historic strains in South Florida and Louisiana and throughout the Southeast. So if there's a silver lining to be had, it's the lack of tubers. I'll just wrap up with this last thought, is the invasive species treadmill. And that's the concept where you get rid of one invasive species and another one takes its place. So that, I don't want to leave on that sad of a note, but ultimately that's what biological invasions do, right? You get rid of one, another one's going to replace it, but that doesn't mean you don't try to get rid of it.
Actually, this was brought in, I think it was on the other side of the pavilion. It's a type of tiger moth caterpillar. We sometimes call them tussock moths because they're very fuzzy. We tell people generally you don't want to handle a caterpillar that's particularly hairy like this. They have what's called urticating hairs, so if they're disturbed, they may actually start to shed those hairs, and they can be very irritating to the skin, especially the eyes, mucous membranes, and the like. So we tell people just leave them be. But this one was brought, they actually broke off a part of a Japanese maple leaf and brought that over to us. We're not 100% sure. We think it might be a hickory tussock moth, but there can be some variability in the caterpillars as they age. But this one is getting ready to pupate. It's quite large, it's quite mature. So what we're probably going to do is let it do that, and then it'll emerge as an adult moth, and then we'll know exactly who it is.
I worked on this guy before I retired. I was the Forest Service, and we worked at Clare Rutledge in conjunction down in Southport, counting egg masses. But I imagine they're everywhere now down there.
Yeah, well, what they're doing, they're being introduced along the I-95 corridor, particularly at truck stops. So the truck stop in Milford is really the epicenter. They're going right along the highways.
And they're looking for the tree of heaven, they're mining those out. We found them, the first one, I don't know about the first one, but one of the local ones in this area was behind the rest area on the Mirror Parkway in Orange.
Yep.
We were able to collect a lot of egg masses behind there.
Yep.
Yeah.
That means they're established.
Yeah, probably came through somebody's car or something, right?
Yeah, and highways are the way these things have been distributed. And Pennsylvania has had a really serious issue, so they come up from Pennsylvania and they've been shipped here. So you just follow the highways, and then they'll go out from the highways.
But I was reading, they're not as killing as like gypsy moth took a lot of, it was massive. But this apparently isn't. It's kind of more of a selective thing, stand-wise.
They are multi-hosts.
Yes.
Because basically you could almost call them giant aphids.
Yes.
Because they have piercing sucking mouthparts. The big issue here in Connecticut is the grape industry. Grapes are fairly sensitive. So what they do is they will tap into the vascular system of the grape, pull the nutrients, denying the grapes reserves, and so then the grapes can't survive through the winter, and then they'll die the next spring. So they will kill grapes.
That's all from this edition of Coast and Country and Plant Science Day 2024. If you want to learn more about the services and resources of the Connecticut Agricultural Experiment Station, then you can visit their website at ct.gov/CAES. Thanks for watching. We'll catch you again next time.